Okay, hey, this is my protein project. The protein that I was assigned is K-Ras, also known as K-Ras in all caps. You can't really see it, but that's written right there. My mutation is glycine AA number 12, which is the original, replaced by aspartic acid, which is the mutation, or cysteine G12D or C. This mutation occurs in 10 to 13% of cancers producing HRAS and NRAS. By the way, that's written right there, but you can't really see it. So I'm gonna turn it around so y'all can see. Pipe cleaners, as you can see, I think, it made the coily shape because it has like a coil spirally shape, okay, Raz. And yeah. And the pom-poms in the front, I use those to make, I use those to represent the amino acids. This is the nucleus because that's where it's located, which is also a limitation because the amino acids is a limitation as well because it's way more than this. So I couldn't represent that because it's way more than this. And as far as the nucleus, it's in the nucleus, but you can't really tell that. Like from looking at this, if I didn't write nucleus, that's why I wrote it. You wouldn't be able to tell where it's actually located. And yeah, other limitations, I can't even show the structure in detail either. Like it was hard for me to show it in detail. I did my best, and you can't really identify like the proliferation differentiation process in the original K-Ras. And like I said, the location isn't really clear. Like you can't really tell. Anyway, moving on. K-Ras is a signal transducer because it's part of a signaling pathway known as the RAS-MAPK pathway, which is represented by the coils, pipe cleaners. The spiral shape, that's the RAS-MAPK pathway. It relays signals from outside the cell to this represents the original. This represents the mutated, if y'all can't tell. That's why I have it written right here, but y'all can't really see. It relays signals from outside the cell to the cell's nucleus using the amino acids, boom, signals. These signals instruct the cell to grow and divide, proliferate, or to mature and take on specialized functions, differentiate, which I said, limitation, you can't really tell. The K-Ras protein is a GT-PACE, which means it converts a molecule called GTP, which is right down, I'm pointing to it, right down here, into another molecule called GDP, which is why I have it written on both sides, if y'all can see. I got GTP on this side, I'm not going to turn it all the way, GDP on that side because it turns, GDP is off, GTP is on, and the original K-Ras can turn on and off, which I have it on both. I have it on both sides, so to represent that it can be turned on and off. And that can bind to both GDP and GTP. These signals instruct the cell to grow, divide, proliferate, or to mature and take on specialized functions, differentiate. The K-Ras protein is a GT-PACE, which means it converts a molecule called GTP into another molecule called GDP. In this way, the K-Ras protein acts like a switch that is turned on and off by the GTP and GDP molecules, like I stated. To transmit signals, it must be turned on by attaching binding to a molecule called GDP. Yep, right there. And the carrying protein is turned off, inactivated, when it converts the GTP to GDP, which is why I have it on both sides to show that it can be bound to GDP or GTP or turned off and turned on. When the protein is bound to GDP, it does not relay signals to the cell's nucleus. K-Ras is normally found in the nucleus of the eukaryotic cells in humans, which cannot be represented. Limitation. The main functional groups present in the original AA side chain of K-Ras are the carboxyl and amine groups, these amino acids. K-Ras is hydrophobic, polar, negatively charged, and is large compared to the other 19 standard AAs. The side chain of K-Ras interact with other hydrophobic areas of the cell due to the farnesylipid side chain. Due to K-Ras's composition of glutamine and negative charge, it can make hydrogen bonds. Glycine AA number 12, replaced by aspartic acid, or cysteine G12D or G12C, makes K-Ras more active, which is why the pipe cleaners are more complex, and I made it more complex and look bigger because it is bigger than the original K-Ras. The mutated version is bigger than the actual original version, and it's more complex. I made it look more complex because of the fact that it's just on, it can't be turned off. It's on, just active. I made it look active. Trying to explain it. It makes K-Ras more active. K-Ras is involved in the signals passed between cells that control the amount of growth that is allowed at any time. K-Ras G12C creates a form of the protein that is always on, like I said, which means this is bound to GTP, just GTP. There's no both sides. It can be turned on and off. It's literally just bound to GTP. It's not bound to anything else, which is why it's always on. This is a disaster because the mutated K-Ras continually tells the cancer cells that it is okay to multiply without the normal limits that control cell growth. The main functional groups present in the original AA side chain of K-Ras G12C are the carboxyl and amine groups, these amino acids, which are bigger because they're more active. Well, not more active, they're only active, which is why they're bigger as well. K-Ras G12C is hydrophobic polar, negatively charged, and is large compared to the other standard AAs. The side chains of K-Ras interact with other hydrophobic areas of the cell due to the farnesylipid side chain. Due to K-Ras's composition of glutamine and negative charge, it can make hydrogen bonds. However, if it is changed to K-Ras G12C, there will be a change in the hydrogen bonding patterns. K-Ras relays signals as far as like how it affects the organism, cell, and all that stuff, the mutation. K-Ras relays signals from outside the cell to the cell's nucleus, right? These signals instruct the cell to grow, divide, proliferate, or to mature and take on specialized functions to differentiate. But if it's converted to K-Ras G12C, it creates a form of the protein that is always on, continually telling the cancer cells that it's okay to multiply without the normal limits that control cell growth. So it's allowing the cancer to grow and spread and decreasing the functions of the organism and increasing their personal mortality rate, meaning like they're more likely to die. Most likely will die, actually. So, yeah. That's my project. Protein project here. You can see a full. I might give you a little 3D for the end. It's the back again. If you want to see how like it's formed, if you can really see it. GTP, GDP molecules. Boom. Thank you.